Hello students, welcome back to the science class. Continuing our lesson about the genetic technology, today we are going to learn about the electrophoresis and polymerase chain reactions. So without further ado, let's begin our lesson. Gel electrophoresis are divided into two types. First, DNA electrophoresis. Second, protein electrophoresis. Electrophoresis consists of two words, electro and phoresis. Electro refers to the energy of the electricity and phoresis means to carry across. So, electrophoresis is a method for the separations of charged macromolecules like DNA, RNA, proteins, and their fragments based on their size. And here, the gel is used as a medium for the macromolecules to move. That's why it's called gel electrophoresis. In this segment, we will talk about the gel electrophoresis of DNA. The basic principle of gel electrophoresis is running a current through the cell loaded with the macromolecules of our interest. Based on their size, the molecules will travel through the gel at different speed, so this will allow them to be separated from each other. It can be based on their charge too, if there's any difference in charge among them. But in case of DNA, all DNA molecules have the same charge to mass ratio and they are negatively charged. Separations also depends on the compactness of the DNA. The more compact the DNA, the faster it will bind to the gel. Before getting into this process, let's learn about the gel first. The gel is a jelly-like material which is composed of tiny molecules and they are held together by hydrogen bonds to form sieve-like structures with pores. Within the gel, the larger and less compact fractions of DNA move slowly, while the smaller and more compact fractions move quickly through this. Two types of gels are commonly used in the separations of DNA. The first one is polyacrylamide gel, and the second is agarose gel. At one end of the gel, some wells are made to load the DNA samples. At first, the gel is placed into the box. One end of this box is hooked to a positive electrode and another end to a negative electrode. Then, the box is filled with a salt-containing buffer solution which can conduct electricity. The end of the gel with wells will be placed near the negative electrode. And being negatively charged, the DNA will move towards the positive electrode from this negative electrode side. To track the movement of DNA, a tracking dye is usually mixed to those samples. Because DNA is nearly transparent, so it can't be observed inside the gel. Bromophenol blue and silene cyano are used as tracking dyes. The tracking dye will always run a little ahead of the smallest fragment of DNA. Also, a DNA staining dye is generally mixed with the gel to visualize the separated DNA fragments after the run ends. To know the size of the different DNA fragments, in the sample we need to run a DNA ladder in one of the wells. The DNA ladder is just a DNA sample, but its fragment size is divided. So, after the run completes, the size of the DNA fragments in the samples can easily be calculated by comparing it with a DNA ladder. After the completion of the run, the gel can be taken out and DNA can be visualized by placing it under UV light. Now, we're going to talk about the functions of DNA electrophoresis. The gel electrophoresis of DNA can be used to estimate the size of DNA fragments following the restrictions enzyme digestion. It can also be done for analyzing the PCR products. Or it can be done to check the match between the DNA samples collected from the crime spot and the DNA of suspected individuals. Now, let's talk about the other type of gel electrophoresis, which is protein electrophoresis. Protein electrophoresis is also known as SDS-PEG or sodium dodecyl sulfate polyacrylamide gel electrophoresis. It is a method that separates proteins based primarily on their molecular weight and electric field by using a discontinuous polyacrylamide gel as a support medium and SDS to denature the proteins. In SDS-PEG, proteins are separated in polyacrylamide gel based on their molecular weight. For sample preparations, a loading buffer containing SDS, beta-mercaptoethanol, bromophenol blue, and glycerol is added to the protein samples. When the sample buffer has been added, the samples are then heated at 95 degrees Celsius for 5 minutes. Proteins are large biomolecules consisting of one or more long chains of amino acid residues. Amino acids are organic compounds that contain amino and carboxyl functional groups along with side chain. Proteins are formed by linking amino acids with peptide bond and fall into one or more specific spatial conformations driven by a number of interactions such as hydrogen bonds, hydrophobic interactions, disulfide bonds, and ionic bonds. SDS is an anionic surfactant and contains a polar hat group with a net negative charge at the end of a long hydrophobic carbon chain. SDS denatures the native proteins by disturbing the hydrogen bonds, hydrophobic and ionic interactions, while the reducing agent beta-mercaptoethanol is used to cleave the disulfide bonds. Also, when the protein mixture is heated, the SDS binds uniformly to the proteins and the intrinsic charges of this protein become negligible when compared to the negative charge contributed by SDS. This treatment brings the folded proteins down to the linear molecules with net negative charge. Therefore, these proteins can be separated in a polyacrylamide gel based on their molecular weight. Positive electrode located at the bottom of the gel, whereas negative electrode positions at the top of the gel. The gel is inserted into a chamber, and the trace glycine chloride buffer systems with pH 8.3 is poured to allow the conduction of current. Through the gel, SDS is also present in the gel and in the running buffer to make sure that once the proteins are denatured, they stay that way throughout the run. For sample applications, in addition to the samples, a molecular weight size marker is usually loaded onto the gel. This consists of proteins of known sizes and thereby allows the estimations of the sizes of the protein in the actual samples which migrate in parallel in different tracks of the gel. Each sample is pipetted into its own well in the gel. And as we have seen previously, bromophenol blue and glycerol are present in the samples. Bromophenol blue is a dye that is useful for visualizing the sample in the well, and glycerol increases the density of the sample and used to make the sample fall to the bottom of the sample well, rather than just flow out and mix with the running buffer. After the sample applications procedure, an electric field is applied across the gel, causing the negatively charged proteins to migrate across the gel away from the negative electrode and towards the positive electrode. Also, small proteins migrate relatively easily through the mass of the gel, while larger proteins are more likely to be retained and thereby migrate more slowly through the gel. Now, let's talk about polymerase chain reaction or PCR. Polymerase chain reactions, or PCR, is a technology indispensable to genetic and molecular biology. Many of its applications are widely used in the world around us, including DNA sequencing, DNA fingerprinting, forensics, detections of microorganisms, and diagnosis of hereditary disease. PCR allows fast and inexpensive amplifications of DNA fragments because it is able to generate large quantities of DNA from a small amount of nucleic acid. PCR is also referred to as molecular photocopying. PCR depends on the series of 20 to 40 repeated cycles of DNA replications by a DNA polymerase enzymes. After each cycle, the number of DNA strands is doubled, and at the end of the 40 cycle reactions, more than 1 trillion copies are generated from a single copy of DNA molecule. The PCR process is separated into 4 steps: initialization, denaturation, annealing, and elongation. In the first initialization step, the reaction is heated to 94 to 96 degrees Celsius for 2 to 10 minutes to activate the DNA polymerase and to denature other contaminants in the mixture. In the case of colony PCR screening, this step also facilitates the cell lysis to release DNA and denature other cellular proteins. In the denaturation step, hydrogen bonds between the double strand of DNA are broken by heating the reactions to 94 to 98 degrees Celsius for 20 to 30 seconds. After the DNA strands are separated, primers bind to a complementary sequence in the DNA template to guide DNA polymerase replication. In this step, the reaction's temperature is lowered to 50 to 65 degrees Celsius for 20 to 40 seconds to allow for optimal primer annealing. After the primers establish a starting point for the enzyme, DNA polymerase start to incorporate the NTPs in a 5 to 3 directions to synthesize a new DNA strand. The temperatures and extension time for this elongation step depend on the type of DNA polymerase enzymes and the target amplicant. Commonly used stack polymerase polymerizes at a speed of 1 to 1.5 kilobases per minute and works ideally at 72 to 78 degrees Celsius. The cycle then repeats from denaturations to elongations 20 to 40 times. At the end of the last cycle, there is a final elongation step that keeps the reaction mixture at 72 to 78 degrees Celsius for 5 to 15 minutes. This ensures that any remaining single-stranded DNA is fully extended after the last PCR cycle. A final holding step keeps the PCR at 4 to 15 degrees Celsius for an indefinite time, keeping the products for short-term storage. Many factors can interfere with a PCR reaction. Some are easy to eliminate while others are trickier to manipulate and will require experience to tackle. In general, factors that are important in PCR include the nucleotide compositions of the DNA template, DNA polymerase enzyme choice, buffer components, primer design, additives, and inhibitors. I think that's all about our lesson today. Don't forget to do the exercise on Edmodo. See you!